Yeah.
Like so.
Okay, let's get started.
What? Yeah. Oh my lord.
Yeah, that's true. We're getting started.
Yeah. Did you get that on camera this time? Oh no. You have to like cut that
That was
out.
Max, by the way.
No, don't.
So, remember, tomorrow, well, tonight at like midnight, the quiz is going to be available, and it's going to cover content from Chapter 4 in the textbook, and Chapter 4 in Elephants on Acid. And Chapter 4 in Elephants on Acid is about sleep experiments. Please don't miss that. You have any time on Thursday to take it.
I love that book, honestly. It's so funny.
Okay, so we left off talking about the cytoskeletal system. Remember, this is a little bit different than the endomembrane system. The endomembrane system consisted of membrane-bound organelles, endoplasmic reticulum, both the smooth and the rough. We talked about vesicles, we talked about the Golgi apparatus, we talked about lysosomes and peroxisomes and things like that. So now we're going to talk about kind of the structural support of the cell and cellular movement. And that's all accomplished by the cytoskeletal system. So there are three elements within the cytoskeletal system that I'm going to want you to be familiar with. Each one of the three has its own protein subunits. So think about cytoskeleton, right, as a series of bones inside the cell. And each of these bones, there are three types that we'll talk about, and each bone has a particular type of protein that it's made from. So we're going to start with the smallest of the elements, and that's the microfilaments. We also call microfilaments actin filaments. Those are synonyms. We call them actin filaments because these microfilaments, their protein subunit from which they are constructed is called actin. So we have microfilaments. Their protein subunit is actin proteins. And these actin proteins occur in couplets, right? There are two of them that occur together. And the way that they connect and are constructed, it causes the actin filaments to form a helix in space. So they actually spin through the cell. Now the function of the microfilaments is cellular movement, a particular type of movement called amoeboid. Yeah.
Is this the first cytoskeleton slide, or is this the second one?
This should be the first.
Okay, sorry.
That's just fine. So the function is movement of the cell, and it's amoeboid movement. And when we talk about amoeboid movement, really what we're talking about is movement of the cell membrane. And so inside our cells, there is a network of actin filaments that goes around the whole surface area, the circumference of the cell. And this network of actin filaments causes the cell membrane to move. So we call that movement amoeboid movement because there are amoebas that move by extending out parts of their plasma membrane and then oozing into that projection. So this is amoeboid movement. So that's for a unicellular organism. Our cells also have, and we're not unicellular obviously, our cells also have actin filaments, and we use actin filaments to divide our cells. So we're going to talk about mitosis and cytokinesis a little bit later. But when our cell organelles have completely replicated themselves, and we have two of everything, and we want to make that one cell two, actin filaments facilitate the pinching off of the plasma membrane in between those two daughter cells. Okay, so really when we think about amoeboid movement, and the type of movement accomplished by actin filaments, you really need to think about it in the context of moving the plasma membrane around. Okay? Okay with that? So that's accomplished using some motor proteins. Oops, 2, 3. So the motor proteins, it's a protein called myosin. And we'll talk a little bit more about myosin and the actin proteins, the actin filaments, in just a few minutes. But for right now, understand that all movement accomplished by actin filaments is done in tandem or in conjunction with myosin motor proteins. The second type of cytoskeletal element are called the intermediate filaments. So the intermediate filaments are a little bit larger than the actin filaments, and they're smaller than the largest of the filaments. And their subunit is called a tetramer protein subunit. Does anybody know what tetra means?
What? Tetra?
Tetra? That prefix, tetra? It's close to 3. 3 plus 1. 4, yeah. So tetra means 4 in this context at least. So a tetramer protein subunit is 4 proteins that are connected together that form kind of like a cuboidal structure. So that says subunit, tetramer protein, the tetramer protein. Their function, however, is not movement at all. Their function is structural support. Structural support. The purpose of the intermediate filaments is to hold things in place. So there's some organelles that we want to move around. Like it's totally cool for vesicles to move around. There are others, however, that we want to stay put. A good example of an organelle that we don't want moving about a whole bunch is the nucleus. We want the nucleus to stay where it is. It's in an optimized position so that it can communicate equally with all areas of the cell. Think about the cheek cells that we looked at yesterday in laboratory. Generally speaking, the nuclei were right in the middle of those cells that we looked at, right? So that's an optimized position, again, for communication with all the different regions of the cell. We don't want it to drift too far to the right because it's going to be too hard to communicate with things on the left. The way that we hold it in place is by creating a scaffolding around it of these intermediate filaments. So it holds things in place, okay? So since it's not facilitating movement, the motor proteins are none. We don't have any motor proteins. Okay, it's not accomplishing movement. Then the last filament is called the microtubules. And it's really easy to get microtubules and microfilaments mixed up, but they're quite different. The microtubules are the largest of the cytoskeletal elements. So microtubules are created from a protein subunit called tubulin. And there are 2 different types of tubulin. There is an alpha and a beta type tubulin. And it causes these microtubules to form helical tubes. They're actually hollow in structure. So the subunit is alpha and beta tubulin subunits. Alpha and beta tubulin. The function is intracellular movement. So when we want to move stuff about inside the cell, we do it on highways created by microtubules. So for example, when we want to take a protein from the endoplasmic reticulum to the Golgi apparatus. We create a vesicle around it, and then that vesicle attaches to a microtubule, and then it rides along the microtubule until it gets to the Golgi apparatus. And then when it leaves the Golgi apparatus and goes to the surface of the cell, another vesicle rides along a microtubule until it gets to the surface of the cell, and then that product is released. So the function is intracellular movement. Now, there are two different types of motor proteins associated with microtubules, and it depends on which direction things are heading. There are things called dyneins, and there are things called kinesins. So for right now, I'm just going to write up motor proteins and then say dynein and kinesin. Dynein is spelled D-Y-N-E-I-N, I think. And kinesin is K-I-N-E-S-I-N. Okay, so in addition to intracellular movement, the microtubules are also responsible for moving large structures like flagella and cilia, excuse me. So you guys saw in the pond water, I hope, a whole bunch of little tiny things flitting around really, really quickly in the water. Those were ciliates. And they're called ciliates because the whole surface of the cell, they're unicellular organisms, are covered by cilia, which are like little oars that paddle them around. And cilia are really, really good for directed, intentional movement. Flagella aren't as good. But it's the microtubules that facilitate that type of movement. Okay, so now we're going to talk about where it is we grow the cytoskeleton from, at least part of the cytoskeleton, one more type of organelle, and then about cell movement. So there is a structure that's part of the cytoskeletal system inside our cells from which microtubules grow. And that structure is called the centriole. The centriole has a unique construction. It's actually membrane bound, but it consists of microtubules in what's called a 9-3 arrangement. So the centriole looks something like this. It has kind of like, think of these as like barrels. It has a barrel that's oriented perhaps directly up in this direction, and then a barrel that's laying over on its side in this direction. And then that centriole, those two barrels, are surrounded by membrane. And it's from this structure that microtubules, some really important microtubules, grow. You guys have already talked about, I know, mitosis in your high school biology classes. Remember, what happens in mitosis is the nucleus dissolves, right? And then the chromosomes all kind of like line up at the middle of the cell, and then you divide up your chromosomes. That's accomplished from this structure. There are two centrioles that grow out microtubules that attach to those chromosomes and cause them to align at the center of the cell, and then pull sister chromatids apart from each other. If you were to take one of these barrels and you were to take a cross-section of it, you would see this 9-3 arrangement. What that means is there's actually a series of 9 microtubules like that. So there are 9 aggregates of microtubules, but the microtubules aren't isolated. They're actually occurring in triplets, like that. So each one of these is a triplet. Like that. And then those triplets are connected with a protein that goes from the top of one triplet to the bottom of another. So it has a star-like or a stellar appearance. So this is a 9-3 arrangement, right? Because there are 9 triplets. So again, that structure is taking one of these barrels, taking a cross section of it, and looking down the barrel, right? Okay, does that make sense? So the centriole is also part of another structure called the basal body. And the basal body is the structure from which a flagellum grows. So we're going to talk a little bit about sexual reproduction a little bit later in the semester. But as it turns out, all the organelles that we inherited from our mother. All of our mitochondria, all of our Golgi apparati, all of our vesicles, we all inherited those things from our mothers. Entire cytoskeleton, all our mothers. The only thing that our fathers contribute to that is 23 chromosomes and a basal body, our original centriole. Okay, we okay with centriole so far? All right. So the last structure that we need to talk about, that's really a part of the endomembrane system, is something called a vacuole. But we talk about vacuoles in the context of cellular movement because in animal-like cells, that's primarily what they're used for. In plant-type cells, they're used very differently. But in animal-type cells, we use vacuoles for movement. So a vacuole is just a membrane-bound sac that holds stuff. In plants, there is a large central vacuole that holds water. And that water is really important because it's the pressure inside that central vacuole exerted against the cell wall that causes a plant to be rigid and stand up. When a plant becomes flaccid, and it's like when you leave your celery in your crisper drawer for too long and you pull it out and it's kind of floppy, that's because the central vacuoles inside the cells of that stalk have become dehydrated. If you were to put that stalk back in some water, it would become rigid again. So vacuoles inside plants are primarily for storing water and other compounds. In animal-like cells, we use them for movement. And when I say we, it's really